thank you very much. I'd like to thank CrossFit for supporting, and Greg, and also like to thank Jeff Glassman for the good questions that he asked us in the past to validate some of our theories. You know, we need people like that. Rather, it's good to have people that question the information that you present. It makes us better at explaining this. So let me go to the next slide here, if I can. Okay, no financial disclosures. All right, so what I'd like to do to start this off is basically to present a report card on our approach to managing cancer. And as I said, I'm going to speak to you today about cancer in general, and also focus on specific types of cancer, in particular glioblastoma, as kind of an illustrative examples of our approach to managing the disease. So let's look, now these are numbers that we can take from the American Cancer Society, and they publish every year data on I guess the war on cancer, the success that we're having. So I compiled just over the last 5 years, this is 2013 to 2017, and you can see these are pretty sobering numbers. You know, we break them down into new cases, deaths per year, and deaths per day, simply dividing by 365 gives kind of an estimate. And you'll notice that the deaths per day and year are exceeding that of the new cases. Not good. Just to put things into perspective, the population increase in the United States over the same period of time was about 2.9%. So you know, how's this war on cancer going, right? You look at the numbers, you can make your own decision. These are the numbers you don't see on TV, right? You see Opdivo and Keytruda and that kind of stuff, but you don't see the constant increase in deaths per day. So the question we have to ask ourselves is, what's going on here? You know, we're not getting success here. This is a failure of monumental proportions, right? The numbers, these are large numbers. In China, it's 8,000, over 8,000 a day in China dying from cancer. Cancer has already superseded heart disease in China. So, you know, we go out and we raise money for cancer, right? You all know, run, jump. I don't know if you do CrossFit for cancer, but everybody raises money for cancer, it makes them feel good. Nobody asks how much of the money that we raise goes to cancer research, and what's more important, what kind of research are you doing with all that money? The federal government's spending millions of dollars on cancer research. People are raising money, stand up to cancer. Look at, the more money we raise for cancer, the more cancer we get. So you have to say, what is going on here? How do you explain this? And it has to do with a fundamental misunderstanding of what the nature of this disease is. We've been led to believe that this is a genetic disease, and I'll present evidence to say that it's not. And we know that here's a simple cartoon of a cell with a nucleus and mitochondria and cell membrane. So we know there are mutations in the nucleus, but we also know that in the mitochondria there are defects as well. And I'll be showing you data that shows that the origin of this disease is a mitochondrial metabolic abnormality. It's not a nuclear genetic disease. And the mutations that you see in the nucleus are actually coming from reactive oxygen species produced by the mitochondria. So what the entire field has been doing over the last 6 or 7 decades is chasing red herrings. Consequently, you have 1,600 people a day dying from the disease. So the current dogma is cancer is a genetic disease. And this is solidified in this major paper, Hallmarks of Cancer by Hanahan and Weinberg, one of the more highly cited papers in the entire cancer field. And what they say is cancer cells carry the oncogenic and tumor suppressor mutations that define cancer as a genetic disease. And we say it's a dogma because it's presented as if it's an irrefutable truth, something that's, and dogmas are no longer questioned, it's a solidified viewpoint. And if you go into any textbook of biology, biochemistry, or cell biology, and you go to the cancer section, it says cancer is a genetic disease. You go onto the NCI website, National Cancer Institute, cancer is a genetic disease. There's no discussion about anything other than the fact that cancer is a genetic disease. Many of you went to medical school. You probably got cancer is a genetic disease. All the college courses on cell biology. Cancer is a genetic disease. So what this concept has done now is indoctrinated several generations of scientists and physicians into this viewpoint that cancer is a genetic disease. The somatic mutation theory is the foundation upon which the viewpoint of cancer as a genetic disease is based. And basically what the somatic mutation theory says is that, well, we get random mutations that accumulate, and eventually you convert a normal appearing cell into this dysmorphic, malignant, mesenchymal kind of cell. But nobody really knows how many mutations it takes to cause or to be related to the formation of a tumor. Is it 1, 2, 4? You know, this Michael Stratton from the UK says we're going to have 100 million genes going to be found. And looking at the deep sequencing coming out of the Broad Institute and these various places, thousands and thousands and thousands of mutations have been identified. And then they have to label them with different names, drivers and passengers and go-alongs and a whole bunch of stuff going on with going on. And we don't talk about those cancers that have no mutations, kind of like a non-discussed. So where does it all lead to us? Where have we come in this journey to manage cancer? We have now come to these terms: personalized therapy, precision medicine. All of this is based on the viewpoint that cancer is a genetic disease. So you have these kinds of images here. You have this woman staring into a screen, and she's looking at a breast cancer information to see if they possess extra copies of a particular gene, which will be used as maybe a diagnostic or possibly some sort of therapeutic application. Now, to get that information, usually you do needle biopsy. So you have to take a needle biopsy of a particular tissue. In this case, it would be a breast cancer, right? So you stab the tissue. And in the process of stabbing the tissue to get the information that she's looking at on that screen, you have changed the microenvironment of that tissue. You have potentially taken a pre-malignant state, and by stabbing it to get this information, you have now put that person at risk. Now, very interesting. The information that you get for this kind of screen is about $7,200 to do one of these screenings, to get the information that you can look at and say, oh, we have this kind of battery of genes. Now, this would be okay if it had any redeeming value, right? It has no value, it's full information. But you put people at risk for cancer by the very process of taking tissue. The phenomenon is called inflammatory oncotaxis. It's an observer effect. By looking at it, you've changed it. Now, I want to talk to you about the evidence that does not support the somatic mutation theory of cancer. And whenever you challenge any kind of a solidified dogma, you always get the same response. We saw one of these images yesterday. This comes from Nico, Japan. These are the Nico monkeys. I went to Japan, actually. They have carvings of them. They're a little bit different than this. But it's basically you don't want to look at the data. You don't want to talk about it. You don't want to hear about it. Anything that challenges your worldview, I don't care if it's a religion, a political philosophy, a scientific concept, generally you get this kind of a response. I know it's hard. It's hard for people to look at things differently. So what I did in chapter 11 of my book, this is a paper that I wrote a couple of years after to bring up to date more and more of the issues associated with information that does not support the somatic mutation theory. And all I did was take articles from the literature that had been spattered about for years and brought them all together in one group of papers and reevaluated the information from those papers in light of the two competing theories for the origin of the disease. So bring them all together and then look at the data and then say, do the data support, did the data support more strongly one hypothesis or theory over the other? And you come to the conclusion that the somatic mutation theory makes no sense relative to the mitochondrial metabolic theory. And you know what's very interesting about this, when Gary gave his talk yesterday, about the difficulty in reproducing findings. I find it remarkable that these different kinds of experiments were done by different individuals, different tumor types, different protocols, but all coming to a similar kind of conclusion that does not support the somatic mutation theory, but more so strongly supports Otto Warburg's theory of disturbed energy metabolism. So let's just look at a couple of these. And the important point about this paper is don't let anyone tell you what they think about it. You're smart people. I tell people, read the original paper, and you come to your own conclusion. You make your own decision. Don't ask, hey, wait, what do you think of that paper? Oh, how do you like it? No, maybe it's no good. You can't believe how many people take in information second, third hand, rather than going to the original source. Read it. You make your decision. You don't like it, do what Dr. Glassman did. Hit me with 1,000 questions, and I'll be happy to answer them. So let's look at some of the data. Now, this was done by McKinnell and his group, published in Science in 1969. I had the chance to speak with Dr. McKinnell before he passed away a couple of years ago, and we discussed these data at length. This frog has a massive renal tumor on the kidney. It's a kidney tumor, kills the frogs, very aggressive. So what McKinnell and his group did is they took this, they captured the isolated cells from this kidney tumor, and then they took the nucleus out of the kidney tumor and put it into a fertilized egg. The original nucleus of the egg was removed. I don't know if I can see it. So right here. So here's the tumor cell. You take the nucleus that has the tumor suppressors and oncogenes and whatever, and you put it into this new cytoplasm that has normal mitochondria, and you get a tadpole, and the tadpole is shown here. And they looked very carefully. There was no evidence of the signature feature of cancer, dysregulated cell growth, anywhere. Everything looked perfectly normal. The problem is this tadpole could not fully develop into a mature frog. So whatever those problem in the tumor nucleus, it was not allowing the organism to fully mature. So it didn't cause cancer, it blocked development. These findings are inconsistent with the somatic mutation theory, which says that the genes are causing the phenotype of dysregulated cell growth. So another paper, I'm only going to give you a through. I put a whole bunch of these. I could take 2 days going over the experiments, but I'm just going to show you a few of them. Now this was by Rudy Anish and his colleagues at MIT, and Rudy's one of the best, most preeminent developmental biologists. And he took these melanoma, malignant melanoma cells, and he characterized all the mutations in the nucleus of the melanoma cell, and then took the nucleus and made embryonic stem cells and cloned mice from the nucleus of a melanoma. And what he says here, the presence of major genetic abnormalities in embryonic mice cloned from the tumor nuclei provides unequivocal genomic evidence that the mice were cloned from the tumor nucleus, but they did not show any dysregulated cell growth. These findings are inconsistent with the somatic mutation theory of cancer. And there was another series of experiments that were done by Dr. Wang and her group at Baylor College of Medicine, where they swap the mitochondria from one cell to the next. These are much, much more difficult experiments than the nuclear transfer experiments, okay? So they take aggressive, malignant, metastatic breast cancer cells, remove the mitochondria from the cytoplasm, and bring in normal mitochondria from normal cells that don't have cancer, and the oncogenes and the abnormal growth was suppressed. On the other hand, if you took the mitochondria from the aggressive breast cancer cells and put them into an indolent cell, type of low-growth cancer, they exploded into high growth. So you got a very different, the mitochondria are calling the shots, not the nucleus. So what we did to kind of convey and summarize all of these data from all of these kinds of nuclear mitochondrial transfer experiments in this simple diagram, which now is making its way through the web. And what I show here is the green cell, which is the normal cells. Normal cells beget normal cells. They have a normal genome. They have normal respiration. The red cell is the tumor cell. Tumor cells beget tumor cells. One tumor cell gets more tumor cells. They have genetic defects in the nucleus, and they also have defects in the mitochondria. Now, what is the origin of the disease? Is it the defects in the nucleus, or is it the defects in the mitochondria? The nuclear transfer and mitochondrial transfer experiments provide evidence for this. Take the red nucleus and move it into the green cytoplasm, and you get normal cells that behave normally, grow normally, form normal tissues, sometimes organs, and sometimes whole mice or frogs. On the other hand, like Israel and Seyfried did, they took the green nucleus and put it into the red cytoplasm, and in that case, you either got dead cells or tumor cells. You did not get normal cells. These are the exact opposite findings you would expect if this disease were a genetic disease. These are the strongest evidence to date to undermine the gene theory of cancer. The strongest evidence. No one has yet been able to explain how we get all these findings based on the somatic mutation theory of cancer. So if that's the case, why is the field continuing to persist with therapies that are based on a flawed underlying hypothesis? So if somatic mutations are not the origin of cancer, how do we get cancer cells? Well, Otto Warburg described this a long time ago, back in the early part of the 20th century. Cancer cells arise from damage to the respiration. Energy through fermentation gradually compensates for the insufficient respiration. Cancer cells continue to ferment lactic acid in the presence of oxygen. This is called the Warburg effect. And unfortunately, the Warburg effect has significantly confused this field, making it confusing to a lot of people. Because they've said, well, there's some tumor cells that don't show a Warburg effect, therefore they're not using, therefore Otto Warburg must be wrong. Well, myself and some of our colleagues, we propose not only that cancer cells can ferment sugar, but they can also ferment amino acids. And that amino acid is primarily glutamine through the succinyl-CoA ligase step. And this is not well known to a lot of people. And basically, it's the missing link in Warburg's central theory. So the cells are fermenting, but they can ferment not only lactic acid, they can ferment amino acids, and particularly glutamine, and I'll present evidence for that. Enhanced fermentation is the signature metabolic malady of all cancer cells. Now, if we take a tumor, and we look at this tumor, and we separate the cells of the tumor, every single cell in that tumor has a different genetic profile. No two cells in that tumor have the same kinds of mutations. This has been demonstrated over and over and over again. However, every cell in that tumor is fermenting. Now, the question I ask to you, is it more logical to focus on the common problem that exists in all of the cells of the tumor, or do you think it makes more sense to focus on the individual unique differences in every cell in that tumor? Right? I mean, the answer should be clear. Wrong. We focus on the unique individual differences at the expense of the common pathophysiology, and that's what we call the somatic mutation theory of cancer. Consequently, we get 1,600 people a day dying. Now, let's look at this energy. All right, so in our normal situation, this is a cartoon of just the mitochondria. So most of the energy that we get in our body comes from breathing, about 89 to 90% through oxidative phosphorylation, respiration. We get smaller amounts of energy through these ancient pathways of substrate-level phosphorylation in the cytoplasm in the form of glycolysis, and in the mitochondrial Krebs cycle in the form of the succinyl-CoA ligase step. And we all know this is biochemistry, right? So we're all breathing. We're all alive, I think. Any zombies out here? They don't breathe. But the issue is most of us breathe, and when you exercise, you breathe more, and this is where we get our energy from, right? Okay, look at the cancer cell now. Okay, this is the same picture, but you'll notice there's a major shift in where the energy is coming from. Much less energy is coming out of oxidative phosphorylation, and a lot more is coming from these ancient primitive pathways, substrate-level phosphorylation. And you see that we now know, and we're learning more, that a majority of the energy is coming out of the mitochondria, but not through OXPHOS, but through the Krebs cycle. This is the new thing that we're talking about. This is the missing link in Warburg's theory. So but we get a lot of energy from fermentation metabolism. These are called, they can get energy without oxygen. And this is where the cancer cells are getting their energy from. So people say, well, how do we get cancer then? So all we have to do is take all of the data that was published in the cancer field over the last 100 years, and just reconfigure it along with Hanahan and Weinberg's hallmarks. Just take the information and just rearrange the picture. And now we can put together in a more logical way the origin, how we get cancer, and eventually, and once we know that, then we'll know how to manage the disease. It becomes much more clear to do that. So what we have here on the left is the mitochondrion, and people say, how do you get cancer? Well, you can get cancer from any number of different things, all right? Carcinogens cause cancer. You can get cancer if you're exposed to carcinogens. You get radiation will cause cancer. Hypoxia, absence of oxygen can cause cancer. Systemic inflammation, we heard from one of the talks that, Axel was telling us about systemic inflammation and others. Rare inherited mutations. People say, oh, it must be a genetic because, you got BRCA1 and believe for how many. Angelina Jolie has her breasts and ovaries removed from the BRCA1. She's trying to reduce her risk. That's all secondary. It's secondary because those BRCA1 mutation does not cause cancer unless it damages the respiration. And there are people around that have BRCA1s who will never get cancer because the gene is not damaging the respiration. RAS oncogene damages respiration. Hepatitis C, papillomaviruses enter and damage respiration. The older you get, so this was referred to as the oncogenic paradox. All right, this was first pointed out by Albert St. Georgi, who said, hey, there's so many different ways to get cancer, but the common pathophysiological mechanism is not clear. Okay? Well, once you understand it's a mitochondrial metabolic disease, the mechanism becomes very clear. And if you read Sid Mukherjee's book on the Emperor of All Maladies, that one that was on the New York Times bestseller, and it was the Pulitzer Prize-winning book on cancer, he struggles with this. If you read page 285 and 303 in his book, it's just like, this is like, we just can't figure out how you get cancer from all these different things. You get cancer from all these different things because they damage respiration and they form reactive oxygen species, and reactive oxygen species are carcinogenic and mutagenic. So the mutations that you see in the nucleus that everybody's following, these red herrings, are all coming as a secondary cause to the damage to the respiration. They're not the cause of the effects, all right? And then when the ROS come out, they damage further the respiration. The cells are suffocating. Where are they going to get their energy? They have to upregulate substrate-level phosphorylation. So you see the green line going down and the red line substrate-level phosphorylation, which means a fermentation metabolism. So what are they fermenting? They're fermenting glucose and glutamine. Those are the two fuels that are driving up the energy, because without energy, nothing lives, period. Energy is everything. Without energy, you don't survive. So what's happening with these cells is they're shifting their energy away from respiration to a fermentation metabolism using available fermentable fuels. So now we can put together all of the hallmarks of cancer in a more logical way, all linked back to damage to the respiration. So the first three hallmarks of Hanahan and Weinberg are all the result of the cell falling back on its default state, the state that the cells had before oxygen came into the atmosphere some 2.5 billion years ago, where everything on the planet was fermenting. They were fermenting amino acids and whatever else they can get. So, and during that period of time, the cells were in a state of unbridled proliferation, and they would proliferate like crazy until the fermentable fuels in the microenvironment disappeared and they croaked. So, and they throw out all this waste material into the microenvironment, leading to vascularization or angiogenesis, another multi-billion-dollar industry that's all based on indirect findings. Okay, then you say, well, if this cancer cell is starting to suffocate, it should die, right? Yeah, it should go undergo programmed cell death and drop dead. It's called apoptosis. Well, why are they not going under apoptosis? Because the mitochondria control the apoptotic signaling system in the cell. They are the kill switch, and your kill switch is broken, and these cells are now bypassing apoptosis. They're not dying, they're proliferating. So the big dog in this whole thing is metastasis. Okay, I can agree with this. Where do you get metastasis from? Which is ultimately the biggest challenge in managing cancer, is trying to control when it spreads through your body. And now you have to understand the biology of the disease. Once you understand the biology of the disease, you can start putting the pieces of the puzzle together. Here's some blue cells. They're columnar epithelial. They could be in the breast, the colon, or wherever. They get damaged by any one of the provocative agents in the microenvironment. They start entering the default state. They start proliferating. Our body has a sensory system to know what's going on. It looks like an unhealed wound. So we have cells, our immune system, that come into these places to heal wounds, and these are mostly macrophages. So they sense this chemically in the blood. They come in out of the bloodstream, and they go right to these incipient cancer cells, growths of cells, to put out the fire, heal the wound, and then heal the tissue. The problem is they throw out growth factors and cytokines, which are actually stimulatory towards these cells, which lost their growth control because of their fermentation behavior. Now, they're making this worse, and it's the wrong context. Now, what these red cells, our immune cells, do is to facilitate wound healing. They fuse together. They're very fusogenic cells, well documented in the scientific literature. So what's happening then with this continual fusion in this muck, this microenvironment, you're diluting the cytoplasm of the red cell with the cytoplasm of the tumor cell, thereby shifting the immune cells from a respiratory system to a fermentation locked in. Now, these cells are already genetically programmed to enter and exit the bloodstream. You don't have to have this epithelial-mesenchymal transition. Makes absolutely no sense. This is the gene theory explanation for metastasis. This is the real thing, and we have evidence to support that in a number of different ways. So you now have a rogue cell, part of our immune system, that's already programmed to spread through your body. Very difficult. They're already programmed to live in hypoxic environments. Therefore, anti-angiogenic things probably won't work, and they haven't worked. So we now know the biology of the metastatic cell. It's a rogue macrophage. What do they eat? They eat glucose and glutamine. Okay, we know that. So now, if most cancer cells obtain energy through fermentation, what therapies might be effective in managing? Well, one of the things logically is simply take away fermentable fuels and replace the body with non-fermentable fuels. And one of the ways to do that is stop eating. Calorie restriction, ketogenic diets, these kinds of things. What they do is their diet restrictions, they differ from starvation. They maintain normal levels of minerals. They enhance mitochondrial biogenesis. And also, they replace fermentable fuel. You can't ferment ketone bodies. You need good respiration for ketone bodies. So you're going to remove the glucose, lower it down, and raise the ketone bodies, which the normal cells are going to shift over to, and the tumor cells are going to be marginalized. You can't use the ketone bodies. And don't forget, we just heard from Michael about the basal metabolic rate. I do this in the mice. The mice, we give 40% calorie restriction, but that's like water-only fasting in humans, okay? People have to realize that because of the 7-fold difference in basal metabolic rate. So ketogenic diets, a lot of misinformation, misunderstanding. Basically, these are low-carb diets, high-fat diets, but it's the types of fats and proteins that play an important role. I don't have time to talk about that. But basically, you eat these diets in a restricted amount. Ketogenic diet, unfortunately, it was labeled with the word diet, all right? Whenever you put diet, everything becomes like mysterious and things. It's a medicine. Ketogenic diet is a medicine. It's called ketogenic metabolic therapy, and it should be respected as a medicine. And if it's not used properly, it won't work, just like any medicine. Not to say that it will harm you, but if you do eat too much ketogenic diet, you can, in fact, get insulin sensitivity. So we worked in the epilepsy field for years, and we understand how some of these diets can be not as effective as they should be. But the whole strategy is not complicated, all right? If the tumor cell needs fermentable fuel, then you take the fermentable fuel away from the tumor cell, and you transition the body to a non-fermentable status. So you lower the blood sugar that the tumor cells need, and you elevate ketone bodies, which the tumor cells can't use, but the normal cells can. You just simply marginalize the tumor. Now, the tumor needs fuel. It can't live without energy. Where's this getting? It's fermenting. You're taking away a prime fuel. What's going to happen to those tumor cells? They're either going to die or they're going to slow down, and that's what happens. Now, the first person that did this work was Linda Nebling, in a human situation, I should say. She took two little children, hopeless cases, brutalized by the system. If you read her PhD dissertation, you'd be crushed about what they did to these little kids. They surgically mutilated them, gave them massive doses of chemo, radiation, all kinds of stuff. And they gave them up for hopeless. They said, these kids aren't going to live more than 2 or 3 months. She says, can I try a ketogenic diet? She was in nursing, getting her PhD in nursing. Yeah, they said, it's not going to do. They don't have long. So anyway, she rescued both of these kids. Their quality of life improved dramatically. They lived far longer than what was predicted. And it was based on the whole shift of the body's metabolism. And I said, wow, this is unbelievable. This was back in 1995. And I said to my students, I said, we should try some of that with our brain cancer in the mice. And we were building these animal models beautiful animal models of human brain cancers. And we had the CT2A, a neural stem cell tumor. Everybody's excited about neural stem cell cancers. So anyway, we just gave them 40%. Standard diet is a high-carb diet, and the same diet restricted by 40%, which is like a water-only therapeutic fast in humans. And these tumors once started to shrink big time, go down 60 to 85% reduction in size. And we say, geez, this is like, wow, I never saw anything like this before. I'm so powerful. What's going on? So then we analyzed using linear regression analysis, using glucose as the independent variable, and either ketones or tumor weight as the independent or the dependent variables, and glucose as the independent. Each square is an animal on a different diet, and you can see on the left here, as blood sugar goes down, ketones go up, and this is an evolutionarily conserved adaptation to food restriction. Bodies are not getting the carbs. We're going to start mobilizing fats, bring them to the liver, chop them up, make water-soluble ketone bodies, and these are going to go to the tissues. And on the right, the blood sugar goes down, the size of the tumor goes down. The higher the sugar, the faster the tumor grows. The lower the glucose, the slower the tumor grows, right? So the higher the sugar, the faster your tumor grows. The lower the sugar, the slower your tumor grows, right? So if you want your tumor to grow fast, get your blood sugar up as high as it can get, right? So you go to oncology clinics, and you see everybody eating ice cream and cake and candies, right? Don't they read the literature? This has been supported now in human gliomas, breast cancer, colon cancer. If you want your tumor to grow fast, get the sugar as high as it can go. Now, people say, well, this looks wonderful and great, but, we don't understand the mechanism. Bullshit, you understand. We publish so many papers, and so many other people publish papers on the mechanisms by which this works. It's anti-angiogenic, anti-inflammatory, pro-apoptotic. No cancer drug is known that can do this without toxicity, and this can do it. So we and others have shown many papers the molecular mechanisms by which this process works. So when you hear people say, well, it's not proven, there's no, they don't read the literature, nor do they contribute to it. So this woman has this dog with a big mass tumor on his nose, right? Menka. Now, she listens to our YouTube videos and reads our regular papers, right? That's a layperson. She doesn't have any training in medicine or anything. So this dog has this big tumor. She goes to the vet. Oh, yeah, we're going to have to cut it out, and then we're going to give radiation and chemo. About $10,000. Maybe the dog will live 7 more months, but it's going to be sick and blah, blah, the same stuff. She says no. So she goes to the butcher, and she gets the fresh chicken meat with the bones in it, cuts the calories by 40%, throws in some medium-chain triglyceride oil and some raw egg. The dog loses about 5% of its body weight. And you can see the tumor starts to shrink and disappear. And Mink is still alive today, doing fine. This was back how fast the dog responded. And there's many dogs now that they're putting through these metabolic therapies that are doing really well. Of course, the veterinarians who are all over me about this, they don't think they should feed the raw meat to the dogs because of salmonella poisoning. Give me a break, So, you ever see what dogs eat? It's like, give me a break. Salmonella poisoning. So you're looking at this stuff and you're saying, gee, and then you did a YouTube video on this for the dog cancer thing. You got 5.3 million hits. Can you believe this? So the tough people are all out there writing all these giving me all kinds of grief. The hell with them, Okay, now I want to talk about a really serious issue here, glioblastoma multiforme, and this is a really bad one, poor prognosis, and unfortunately Senator John McCain is now struggling with this kind of a tumor. It's a nasty, nasty tumor. Multiple different kinds of cells, no effective therapy. So you get a whole bunch of different kinds of cells, consequently the name multiforme, highly invasive. All right, so when you look at a brain tumor, here's a poor soul that sacrificed their brain for the study. And you can see this nasty necrotic area, large cyst. But if you look at the midline of the brain, you can see it shifted to the left. This is called midline shift. Okay, so these tumors grow and they cause intracranial pressure, and people die from intracranial pressure, most of the people who have these kinds of tumors. And the problem is you can't surgically resect them because the tumor cells have already spread out into the normal appearing size. And the tumor cells, one of the mechanisms they use is they go across the surface of blood vessels in the Virchow-Robin space. So they use these blood vessels as kind of a railroad system to get through the brain. So it's very, very hard to any kind of surgical resection. And you can see them, the dark blue cells around the blood vessels are you can see on histology. Histology will tell you. So this is how they spread through the brain and make it very difficult to get resolution. And we all know that mitochondria are abnormal in brain cancer. And this shows you a picture of an electron microgram. The only way you can see mitochondria clearly is EM, electron microscopy. And the stripes in the mitochondria contain the proteins and the lipids of the electron transport chain that allow us to get energy through oxidative phosphorylation. So you can see the nice stripes on the normal mitochondria there. And the GBM mitochondria, mitochondrial glioblastoma multiforme, is called crystallysis. The very structure of the organelle needed for oxidative phosphorylation is missing. Anybody can see there's an emptiness in that mitochondria. The stripes are missing. The stripes are missing, therefore the oxidative phosphorylation is missing. Therefore the cell must ferment in order to survive. Everybody see that? Okay, there's a lot of papers in the literature showing that, yet many members of my field say mitochondria are normal. They obviously don't look at this, or they don't want to see about it. They ferment. They have to ferment. They don't have the structure. Structure dictates function. Without the structure, you can't get the function. The function is abnormal because the structure is abnormal. And my colleague, Gabriel Arizmendi-Murillo, has published many beautiful papers on this. So if you can't get energy from oxidative phosphorylation, where do you get the energy to drive the beast? Where is that energy coming from? It's coming from glucose and glutamine, the two prime fuels that are going to drive the beast. But they can't eat anything else. It's not there in sufficient quantity. We did the logistics on this. These two fuels are abundant in the microenvironment. So they come in, right? They two together, they're synergistic. Glucose, glutamine, they come in, they fire the glycolytic pathway, the pentose pathway, the glutaminolysis pathway, driving energy from substrate level phosphorylation, making all of the stuff for DNA, RNA, proliferation. These cells grow like crazy. So what do we do in the clinic? Okay, so some poor soul comes in, diagnosed with a glioblastoma, devastating. You know, people don't know what they're, almost like the head. You're just devastated. The patient's devastated. The family is devastated. You know, you got, it's my God, what's going on here? Well, we got to get you in and cut that tumor out right away. Sometimes you have to do that. This is a herniation problem, but many times you don't. You have a watchful waiting period. No, we get them in as quickly as possible. Okay, we're going to debulk the tumor. Surgeon gets a hold, out comes the tumor. Patient's sitting there, wakes up, oh, wow, hey, you know, how do you feel? I feel pretty good. Okay, now as soon as you recover, we're going to start giving you radiation therapy. So in the brain, neurons and glia have a very close connection with each other, intimate relationship, right? It's called the glutamine-glutamate cycle. It keeps our neurotransmitters in balance and everything is under control. You break that glutamine-glutamine cycle, glutamate, which is an excitatory neurotransmitter, comes out, excites neurons, they die, you get necrotic death. Astrocytes take up the glutamate, turn it into glutamine. Now, those tumor cells that have not been debulked will now sucking down the glutamine created by not only the wound from the surgeon, but also the radiation that's blowing the hell out of the microenvironment in this tumor, creating a vast amount of glutamine, which I just said is one of the powerful fuels driving the beast, right? Now, when you take somebody and surgically resect and debulk them and then start radiating their brain, you start to get head swelling, brain swelling, from the radiation causing the heat, the edema. To reduce the edema, we give them high-dose steroids. High-dose steroids create hyperglycemia, right? Glucose and glutamine are now created by the very procedures that are used to treat the patient. Now, to make matters worse, 90% of the brain tumor cells are infected with human cytomegalovirus, which is a supercharger for allowing the tumor cells to use glucose and glutamine. So I published this paper in Lancet Oncology saying that the standard of care contributes to the growth and the recurrence of the tumor, based on hard biochemical evidence. What do you think the response is? You don't want to hear about it. Now, let's test the hypothesis about what I just said. Okay, look at the results from treating patients with brain tumors with the standard of care. So we have two lines here. The red line are those individuals that got radiation alone, and the blue line are those individuals that get radiation coupled with the toxic alkylating agent called temozolomide. And the fact that temozolomide could contribute a little bit to survival was the single greatest advance in glioblastoma management in the last 50 years. Can you believe this? I was at when they said this. Now look at the red line, the guys that got the radiation alone. How many survivors came out of this study? What's that number down at the end there? 0. This has been reproduced. You want to know about replicating data? This has been reproduced in every country in the world over and over again. Nothing is more certain than irradiating people and having them all dead. Now we throw in temozolomide, but hey, listen, temozolomide is doing something. Wow, making you get a few extra survivors from this. So I said to myself and my students, what is temozolomide? So we looked and we found out the adverse effects of temozolomide are diarrhea, vomiting, nausea, and fatigue. Wow, these are all indirect forms of calorie restriction. So we published and we said, we think that blip is due to indirect calorie restriction. How do you think? Do you think anybody ran out to test that hypothesis? No. You know, and temozolomide also increases driver mutations. What the hell does that mean? Shouldn't temozolomide make the tumors grow faster if they increase driver mutations? We don't see that. Now let's put a face on this. Okay, so this is Brittany Maynard. She was a young California girl, Northern California, diagnosed with brain cancer in January 2014. The tumor was a small, low-grade tumor. And within 1 month after taking out the low-grade tumor, it morphed into a glioblastoma multiforme. She was then treated with standard of care, heavy doses of steroids. And you can see her face on the right there. It looks totally different than when she just got married over here on the left. That's called moon face from overdosing steroids, all right? So she says, I'm out of here. She's going to go to Oregon, next town up, next state up, and die with dignity with her family. She's going to throw the towel in. And she put an article out, was in People magazine, about her decision to die. And the People magazine article was all about the morality of death with dignity. Nothing about the miserable failure of the standard of care that put her in that position in the first place. Now, what does it say when your patients kill themselves rather than continue with your therapy? Not good. Now let's look at another face. This is Pablo Kelly. Same age as Brittany, about 28. He's diagnosed with glioblastoma in August 2014. He emails me and says, can you help me? I don't want surgery, I don't want radiation, I don't want chemo. So I give him the kit that I send to most of the cancer patients who contact me. And I said, hey, you might as well try it, Pablo, because I'm definitely not doing it. He's from Devon, England. He says, okay. So I give him the kit. I hadn't heard from him. I didn't, maybe a year and a half go by. All of a sudden, I get a letter from Pablo. I said, oh, geez, Pablo. Pablo, he's still alive. So he went on. He had this big YouTube video telling everybody how he was going to do keto and all this stuff. But anyway, he says his formerly inoperable tumor now has become operable. So he asked, I said, well, if you can shrink it down, get it out, Pablo. So he goes out and it was earlier this year, gets the tumor out. He has a few seizures, but his quality of life is pretty good. His wife just had a baby. He's still alive. He's doing well, right? He has a quality of life. He's alive. So what we did, knowing about all these situations, we built the glucose ketone index calculator, which helps patients and others get into therapeutic ketosis. So if you want to stop the growth of the tumor, the first step you got to do is get into therapeutic ketosis. And it's the ratio of glucose millimolar, ketone millimolar with one of these precision extra keto mojo things. We're testing all these things against chemistry. And it helps patients stop the growth of the tumor. So it makes it easier. Rather than trying to measure the two fuels together, you get a single value. We also built some of the greatest models, the most replicatable models to human glioblastoma, spontaneous brain tumors in the mouse, so you know that they're coming from the host. And you can see this mouse, M3, is a glioblastoma, VM-M3. And on the left here, you can see the tumor. It invades right through the brain using the same mechanisms as you see in human. On the right is the same tumor in mice treated with calorie restriction and ketogenic diets. Hard, we pounded these things. Now, we stopped invasion. You can see there's much less invasive behavior, but we couldn't kill the cells. They're growing. I threw everything at these things. We were fasting these mice. We were bringing the sugars down, ketones up, and the damn tumor cells are still alive. And the humans get much worse. I tell you, I don't understand how the human brain can be. They're so much more receptive to this therapy than the mouse brain is. People say you cure mice all the time. You don't cure mice all the time. You get the mouse that has the same tumor as human, you get the same problem. So I said they must be using glutamine. So we tested the glutamine hypothesis by using 6-diazo-5-oxo-L-norleucine, which is DON. It's been used, it's an old drug. It was used years ago. It worked out for some cancers, not other cancers. You know, it was just one of those things. Some were a little too toxic, some not toxic. Anyway, what it does is it stops glutamine metabolism. All right, I can go through the details. So we decided to test mice with DON. And we decided to put the tumors into the brain and then give them a fast, let them go for 3 days until the tumors are raging, then put them on a fast, and then switch them back either to a high carbohydrate diet or a ketogenic diet restricted, with or without DON. And on the top, then we pulse DON 6, 8, 10, 12. Every other day, give them a little DON, give them a little DON while they're on these diets. We stopped experiments at day 15 because the control guys, they're dying. They're starting to get morbid because the tumors are growing so fast from all the carbohydrates and everything. So we then compare and contrast the brain tissues and the biochemistry of the tumors at 15 days. And we genetically engineered these cells to be bioluminescent so we can see how active the tumor is by putting them into a bioluminescence Xenogen machine. And so we take the brains out of the mice and we put them in these petri dishes and put some luciferin in there, and the light tells us how many living tumor cells there are in the brains of these mice that are treated under these different conditions. And the guys on the top, you see a lot of bright light, reds and yellows and all this. And they're the ones that didn't get anything but the high-carb diet. They're raging. And we have a lot of different studies to show this. The ketogenic diet restricted, you can still see there's a lot of living tumor cells in the brains of these mice. We did not cure these mice with ketogenic diets. They don't invade as much, and I'll show you evidence for that. But when you put the DON, the glutamine inhibitor, together with the diet, we got no light. All right? There was no light. We did this over and over again. And here's another series of individual mice. You know, 1 through 6 guys that got the diet only, and then the other guys, 1 through 8, got the diet with the DON. And that background, that's all background except for one mouse there who had a little breakthrough light. But by and large, we really eliminated the light and the living cells in these tumors by putting the diet together with the DON. The DON by itself is okay, but it doesn't get rid of as much light as putting the DON with the diet. The two together work best. And of course, the gold standard for determining cancer is histology. That's what they do. They take needle biopsies. Oh, you look at the cells, you got cancer, right? So you have to do histology to determine whether or not what's going on inside the tissue. And when we do that, we have the standard diet, the ketogenic diet restricted, and the diet restricted with the DON. Low power, high power is what you're seeing. And if you look at the standard diet, you go, oh, these cells are piled on top of it. They can't grow any faster than they're growing. The mitotic figures, the cells are densely packed, and that's what you get when you have a high carbohydrate diet. The ketogenic diet, the one in the middle, you can see the white part of the brain there. That's the normal part. And it's blocking the invasion from the dark blue into the white. You can see how much the invasion is over here in the high-carb diet. The cells are spaced further apart. So what it's telling us is the diet is preventing invasion and it's stopping the rate of growth, but they're still growing. On the far right, you got the DON and what we see, that is all dead cells, all right? Blasted, slaughtered, these cells. They're all broken, mitotic arrest, necrotic. They're all dead. This supports the fact that we didn't have any light. The tumor cells are dead. So based on this, we developed the press pulse therapeutic strategy for humans based on our preclinical studies in mice. And I worked together with Dom D'Agostino, Joe Maroon, the neurosurgeon, George Yu is an oncologist. And we put this together, and I don't have time to tell you where the origin of press pulse comes. But basically, we use press therapies, which can include the ketogenic diet, restricted ketone supplementation, and stress management. You know, you can't believe that when you have cancer, people get stressed out. You have this impending doom. I'm going to die. What does that do? It raises your blood sugar. Cortisol goes up. You've got to have stress management. So we use exercise. We use music therapy. We use yoga therapy. Whatever works to lower the stress of the individual. We use the diet as a press. The diet is controlling the availability of sugar and raising ketones that the tumor cells can't use. Once we get the patient into therapeutic ketosis and lowered stress, we then apply pulses. And we use drugs like 2-deoxyglucose, insulin potentiation therapy. We then hit them with glutamine inhibitors like EGCG, the green tea extract, chloroquine, the anti-inflammatory, and we're working, and we would like to get DON, of course, and others. And then we put them in hyperbaric oxygen chambers. Hyperbaric oxygen will kill tumor cells just like radiation does without toxicity once you remove the glucose and glutamine, get the patient into therapeutic ketosis. So we can replace the entire standard of care with a logical therapeutic process that's non-toxic. And we gradually move the patient from the disease state to the so-called managed state, and eventually, hopefully, to a long-term management and possible resolution using press pulse metabolic therapy. Now, how does it work? Okay, so here's a paper that we just published on a patient with glioblastoma from Egypt. We can't do this in the United States. You can't do this in England. All right? You can't do it in a lot of places. So we had to go to Egypt, where they read our books, they read our stuff, and they said, okay, we're going to try this. They came to me and told me how they're doing this, and they said, can you help us write it all up? And you know, a lot of physicians can't write papers. I do this for a living, right? So I said, we'll get the data, we'll put it all together just like we do the mice, and we'll set up a protocol. So management of glioblastoma in patients with ketogenic metabolic therapy with modified standard of care. That's what we can't do so far in this country. IRBs will not allow us to modify the standards of care. So we took this guy, he came in, his whole left side, he's dragging. You know, he was a metabolic mess. He had pre-diabetes, he had low vitamin D, he had all kinds of other issues besides he had a glioblastoma. So the first thing Dr. Elsaka did, we gave him a 3-day water-only fast and then transitioned the guy to a 900 kilocalorie a day for 21 days with the ketogenic diet. So he's out over 3 weeks before we touched him. Then he did an awake craniotomy. We debulked the tumor. The tumor looks different now. The tumor has a different morphology based on the treatment of the diet up front. You're shrinking down those cells. Then for another 3 months, we gave him chloroquine, we gave him EGCG, we gave him hyperbaric oxygen. Then we were forced into doing standard of care because they have to do it. So, and while he's getting the standard of care, which is radiation and chemo, he's also on hyperbaric oxygen and the diet as well. And then for another 3 months, you can see after that. And now at 24 months, the guy is doing fine, right? Now he's a corn farmer. He's back out working in the fields. So now he's out 30 months and he's still doing. I just got talked to Osaka the other day. I said, how's the guy doing, the corn farmer? He's doing fine. So he had a little radiation edema, which pisses us all off, something fierce. You don't want to radiate the human brain under any conditions. You know, I don't understand that. It kills me. You know, I'm looking at these poor people and I'm saying, what are you doing? Well, we have to kill the tumor cells. Just take away the glucose and glutamine, you get the same effect. So here you can see that. Look at this. Bring his GKI down, glucose ketone index, got it down really nice. Guy's in therapeutic ketosis. And on the bottom, look at this. Okay, you see the red line on the bottom? You see his big tumor there? Okay, watch as the treatment continues. At the very end, you see the red line is now straight. All right? We corrected the midline shift, and the guy's doing fine. And it's just not brain cancer. This is a patient who had triple-negative breast cancer from our colleagues in Turkey, in the Istanbul clinic, and they're treating all kinds of lung cancer, pancreatic, all stage 4 cancers. This is stage 4 woman, comes in with triple-negative breast cancer. Number 1 is in the breast, 2 is in the liver, 3 is in soft muscle tissue. Again, ketogenic diets, hypothermia, hyperbaric oxygen, the lowest dose of chemo possible to remain compliant with the law. I said to Slocum, I said, what happens if we get rid of the chemo? Patient would do better. But you got to do it, because you're going to lose your license if you don't. Can you believe this? So anyway, this patient is doing well. We published it in Curis Biomedical Open Access. And somebody, oh, how's that patient doing? How's that patient? Well, Slocum says the patient is still doing well, never lost hair, always had a high quality of life, never got sick. And we're seeing this over and over again. Not everybody. I don't want to make it look like, hey, everybody's doing well. You know, there's a few people who don't make it. They've been beat up so bad by the traditional standards of care, their bodies can't rally. They're so demolished in their ability to heal themselves by the traditional standards of care, they can't rally. So we have this thing, GBM and other stage 4 cancers. I don't consider them as terminal cancers. I don't think they should be considered terminal cancers. Okay? Because we're mistreating the patients. We're putting them into risk of death by the very treatments that we're using to try to save them. Makes no sense. You poison or radiate people to make them healthy? Give me a break. You got Brittany Maynard, standard of care. And then you get Pablo, rejects standard of care. Oh, they're only one person. No, there's not one. There's Alison Gannett. There's Andrew Scarborough. There's a whole bunch out there. We just haven't published them yet. So conclusions, cancer is a type of mitochondrial metabolic. It's not a genetic disease, okay? This misunderstanding is the greatest tragedy in the history of medicine, leading to the unnecessary suffering and death of tens of millions of people by a fundamental misunderstanding of what the nature of the disease is. These cells rely on substrate-level phosphorylation. It's the hallmark of what these cells do. They're dependent on glucose and glutamine as the prime fuels for GBM and all these other cancers. They need that fermentable fuel. Who's targeting the fermentable fuels? Nobody. The press pulse metabolic therapy is non-toxic, cost-effective for the management and possible resolution of all types of cancers. It's a singular disease. All these tumor cells are all fermenters. It doesn't make any difference. It's my opinion, and I could be, I don't know if I'll live to see it, that this strategy will eventually make all other strategies obsolete. It's just a matter of time. So I want to thank my collaborators and colleagues from the United States, from Turkey, from Germany, from Venezuela, from Hungary, Greece, France, Egypt, India, and China. The Chinese want to now start dovetailing this into their traditional Chinese medicine. And I'd especially like to thank the funding that we get, which is very hard, believe me. You get massive amounts of money to study cancer. You get very little if you want to try to resolve the disease. Single Cure, Single Cause Foundation, CrossFit, thank you very much. Dr. Joe Maroon, team surgeon for the Pittsburgh Steelers, distinguished neurosurgeon at the University of Pittsburgh. George Yu, oncologist. Ellen Davis, Boston College, and in the past, the NIH. Thank you for your attention.